Thank you all so much for joining us today for the iSeq 100 introduction webinar. I really appreciate all of you taking some time out of your day. All right, so let's go ahead and take a look at our agenda today. So in today's webinar, we will be touching on the following topics. First, we will review the iSeq 100 instrument itself and discuss some of the specifications. Then we'll touch on some of the key technologies that enable the iSeq 100 sequencing instrument to do what it does. Next, we'll go over workflow of both the setup and use of the instrument. We'll briefly touch on a few troubleshooting steps that we have found to be helpful should any issues arise. And lastly, we'll touch on the many resources available where you can learn more on the topics covered today. I know that we may have a broad audience today, with some of us being new to next generation sequencing and others being veterans. This webinar is intended as an introduction to topics rather than a deep dive on any one of them. However, throughout the presentation, I will be providing some links to some really great resources that might go into further depth. And as Dina mentioned, put any questions in the Q&A chat and we can dive a little bit deeper on those particular topics that you're interested in at the end of the webinar today. All right, so we can go ahead and get started on overview and specifications. So this is the iSeq 100. It is the smallest Illumina sequencer at about 1 square foot of bench space, but it's still capable of generating over 4 million reads passing filter. When a run is set up as a 2x150 run, it generates over 1.2 gigabases of sequencing data. The max supported read length is a 2x150, which takes about 18.5 hours to complete, with sequencing of about greater than 80% bases, higher than Q30. Shorter reads will take less time and generate less data. We'll go over what some of those run times might look like at the specifications part of this section. Moving on to some of the key technologies on the iSeq 100 instrument. First and foremost is the CMOS chip and the one-color SBS chemistry. We'll have a chunk of our webinar talking about this. It's a really cool piece of technology. Really excited about this on the iSeq 100. We also have onboard denaturation. So we are actually loading double-stranded DNA onto these cartridges here. It's using pattern flow cell technology that was first introduced on the iSeq 4000. It's really cool to get to see that technology coming to this small instrument to really maximize the benefits of this instrument. It has a really simple user interface. If anyone has used the NovaSeq instrument, it's a kind of similar interface to that, very easy to use and very easy to load. We'll go over a video on how to actually get a run started on this instrument. It's very easy. We also have cartridge-based fluidics, which is huge. That's recently been brought to the NextSeq 2000 instrument. And so we have cartridge-based fluidics, no more doing any washes. And like other benchtop instruments, we also have onboard analysis using local run manager or LRM. And we'll touch on that a little bit at the end of this section as well. To those cartridges, the reagents on this instrument, it really helps contribute to the instrument's overall small footprint. These reagent cartridges are fairly small, and the reagent cartridge and flow cell contain all of the needed reagents, as well as the entire fluidic system. This helps make the setup extremely simple, and samples and reagents never leave the cartridge. It's a really cool tool that allows for really quick setup and cleanup of these runs and ease of maintenance for the iSeq 100. To the reagents themselves, there have been some updates to the iSeq 100. We now have V2 reagents. Those have been available since April 17th of 2020. There are a number of improvements, including improved formulation and recipe, which reduces the risk of clustering failure on the iSeq 100. We've also rounded the cartridge corners to protect that cartridge bag, the foil bag, during thawing. We've also increased the stability of these reagents. So if you thaw these kits in the fridge at 4 degrees for 36 hours, that reagent cartridge is stable for up to 1 week prior to use, which really just helps keep the lab able to use iSeq reagents and making those more available, as these do have a longer thaw time, which we'll talk about later during run setup. And with the V2 reagents, we also have a new version of the control software. We have control software V2. This update The two biggest highlights, I think, are that we can now use V2 reagents. It has the recipes that allow it to run those reagents. And it also extends the number of validated uses from the test kit from 36 up to 130, which is pretty significant and increases the lifespan of that system check kit. And if you have any other questions about the V2 reagents, we have a really great bulletin that I've linked here. And if you have any questions about the iSeq control software V2, there's some more information over on that downloads link there. The iSeq control software V2 is user installable, and so the lab can install this control software at any time. To that point, the standard iSeq 100 I1 reagents are being obsolesced on March 1st of 2021, so right around the corner. The iSeq 100 reagents, the standard version, the last order date is the 28th of February, and then the last ship date of the 31st of March 2021. Did kind of want to call that out here in case anyone needs to order those. But I do really recommend moving to the V2 reagents. The improved formulation here is really critical to getting more frequently good runs out of these reagents. So really excited about those changes made. With the iSeq 100 V2 reagents, we have updated our loading recommendations, which I've listed out here. This recommendation list comes from that bulletin as well as the system guide, but I did kind of just want to bring that up to you all here, that there are some updated recommended concentrations. You can see the V1 reagent guidance on the right there and the recommended loading guidance on the left there. These are some of the libraries that we at Illumina have validated on the iSeq 100. If you're running a different application, the recommendation is to run that library on the V1, to run the library that you've validated on the V1 reagents at the same loading concentration on the V2 reagents, and then optimizing from there. So starting with that V1 and then optimizing afterwards. All right, and then I also wanted to talk about the specifications for these reagents. We don't have to talk through all of these. I wanted this more to be a resource page for you. I think that the specifications, they're all coming from that iSeq 100 specs page, which I have linked at the bottom here. And I just wanted to highlight, the different run times, the different Q30 scores that you can expect from different run setups, as well as the output. When I'm reviewing a run for the first time, I like to keep these specifications in mind when I'm thinking about, is this run good? And so this could be a helpful resource at the lab bench, just to kind of eyeball how are the Q30 scores and output expected to what we should be getting for that library prep, or sorry, for that iSeq 100 instrument. All right, and then the last thing that I wanted to talk about on the iSeq 100 system is this test cartridge and test flow cell, which together are the reusable test kit. Each iSeq 100 comes with a test kit that is validated with that instrument. And it first, enables you to install the instrument in the lab. It can be used as well whenever you need to test the system for any troubleshooting. And it even allows you for some reinstallation. The iSeq 100 instrument is very small and doesn't have sensitive components that can be jostled around when moved. And so this instrument can be moved by the lab if there's a more convenient place for it. After moving it, just to make sure that it's still running well, you can go ahead and run the system check on it, just to ensure that all of the components are still running there. And again, with that latest version of the control software, these test kits are good for 5 years or 130 uses. So I wanted to highlight that as well. All right, so that was an overview of the instrument itself. So now let's dive into some of the technology on this instrument. And the first thing that I want to talk about is the flow cell architecture. I kind of mentioned it when we talked about the pattern flow cell being on the iSeq 100. But just for a little Illumina history, we have both non-pattern flow cells and pattern flow cells. The non-pattern flow cells are on your instrument, like the MySeq instrument, the NextSeq 500 instrument, and it is essentially an open oligo lawn where clusters are randomly spaced and can cluster anywhere on the flow cell. With the pattern flow cells, we're actually letting the letting the DNA cluster in certain nanowells, and they are optimally fixed cluster spacing so that we can maximize the cluster density on these flow cells. It's one of the ways that we're able to get 4 million reads from such a small amount of area on this iSeq 100 chip here. And so just to kind of show what that cluster generation looks like on these pattern flow cells, you can see those nanowells here represented in like this hexagon lawn here. And so you can see one strand of DNA that has landed in one of these nanowells. And the way that this pattern flow cell technology works is I've had it described to me kind of like popcorn popping, is that once that DNA seeds that well, it pretty instantly clusters, forming a monoclonal template of that DNA. And this is happening across the millions of nanowells on the flow cell. And so just a really cool piece of technology that we're bringing to this smaller instrument here. And if you have any questions about how pattern flow cell technology works, this is probably like my second favorite YouTube video resource that we have, this pattern flow cell technology video. It's really helpful, second only to the SPS chemistry video, which is also another great one that we have. So moving right along to the sequencing by synthesis. So all of our Illumina sequencers utilize sequencing by synthesis, which is essentially incorporating one base at a time, where we have base detection is completed, and then after that we go ahead and incorporate the next base. If you have any questions about SPS chemistry, we have a really great webinar that dives really deep into the general basics of SPS chemistry and how it performs on the different instruments. We'll go over a quick little overview here when we're talking about the SPS chemistry on the iSeq 100. So at first, Illumina developed the 4-channel chemistry with 4 fluorescences, 1 per base. Next, we had 2-channel chemistry with 2 fluorescences and some image comparisons that enabled us to make 4 base calls. And then the new aspect of SPS chemistry for the iSeq 100 is a 1-channel chemistry. And we have 2 chemistry steps performed for each cycle of sequencing. Similar to 2-channel technology, 2 images are taken for each cycle, but instead of the 2 images being taken in 2 different colors, it's being taken in the same color or the same channel and just taken at 2 separate time points. In between the pictures, the fluorescence molecule attached to the A base is going to be cleaved off and a new one attaches to the C base. The T base has a fluorescent molecule that is unaffected by the chemistry cycle, and the G base does not have a fluorescent molecule attached at all, similar to our 2-channel chemistry here. This leads to the instrument being able to make 4 base calls from a single light source. And we have some more information about that CMOS chip and 1-channel SPS chemistry linked here. I have that paper actually linked a couple times here coming up, just because it's such a great resource for us. And so the pattern flow cell technology is critical to letting the iSeq 100 instrument be able to do what it does. So other Illumina instruments use a classical optical system that uses cameras focusing on a flow cell similar to a microscope. The iSeq 100 instead uses a CMOS sensor paired with a flow cell in a 1-to-1 pixel to cluster ratio. So underneath every single one of those nanowells is a CMOS sensor that is able to detect the presence of light emitted or the lack of presence. So those CMOS sensors are those digital image sensors similar to those used in cameras. And because they're underneath each cluster there, you don't need any lens or sensitive optical components. And then that image, and then we have here just an image of a cross section of what that looks like. We have that single color excitation light, the flow cell glass, and then we have that CMOS photodiodes at the bottom there that allow us to actually capture the presence or lack of presence on the flow cell. Because we have that CMOS chip here that helps actually take the pictures for us, all of the flow cells are considered electronic waste at the end of a run, and we'll touch on that a little bit later. But each of these flow cells does have an electrical chip on it. And then, so just one more time, really breaking down the base calls. All over the flow cell, we have those CMOS sensors that are looking for intensity in each image taken with those intermediate chemistry steps, changing the picture slightly and allowing us to make base calls. Again, I just wanted to add this information here just because I think that it's really cool to see how this technology works and how it makes those base calls. And again, that information lives on that CMOS chip and one-channel SPS chemistry note here. And last, I wanted to talk about Local Run Manager. This last piece of technology really enables the iSeq 100 to be a complete solution in a small box. Local Run Manager, or LRM, is a software package that handles user management, run tracking, secondary analysis, and allows us to view some preliminary results from a sequencing run. It can be accessed directly on the instrument or remotely. It's a really great tool. I know a lot of users use it just to generate fast queues, but there are a lot of other modules and analysis pipelines that can be run right on Local Run Manager. And I have linked here a couple of trainings on using Local Run Manager, as well as some documentation on Local Run Manager as well, if you're interested in looking at those. All right, so we've gone over the instrument and some of the key technologies. So let's look at what it's like to actually run an iSeq 100. So kind of at a glance, these are the different steps that we have to starting a run. The very first thing you should do, again, because these reagent cartridges do take a while to thaw, is to start thawing your reagent kit. Then you'll go ahead and dilute your library and load it onto the cartridge. Notice here that we're not doing a denaturation step. With the iSeq 100, we are actually loading double-stranded DNA onto that cartridge. And then the cartridge will do that denaturation for you. After we've loaded our cartridge up, we're going to go ahead and set up our run on the instrument, whether that's through Local Run Manager or through a manual run setup. Then we're going to go ahead and start the run, and then you can monitor that run on Base Sequence Hub or Sequencing Analysis Viewer, or on the instrument itself, just to see how that run is looking. About 5 hours after the run starts, you should start to see metrics such as clusters passing filter and Q30 scores. All right, so to start, we have our thawing. Cartridge thawing is done in the foil bag out of the box. Before starting the run, we also recommend bringing the flow cells to room temperature. It takes about 15 minutes for that to happen, and I recommend doing that before loading the instrument. And then one note here is to not move that reagent cartridge straight from a shipment of dry ice to a water bath. That can shock the reagent cartridge, and there are some sensitive components on that reagent cartridge that can cause some errors. And so do recommend, letting that reagent cartridge sit at 4C overnight sit in the freezer overnight before starting the thawing process for that. If you want to use the water bath option, you can set it to room temperature in the ambient air or at 4C. And so thawing these iSeq 100 kits, the first thing you'll notice is that this does take longer than some of the other Illumina sequencing kits. And that's because these cartridges are more complex than other Illumina reagent kits. To start, we have those reagents inside plastic tubes, which are surrounded by air, which is surrounded by the cartridge housing, more air, and then the foil bag at the very end there. The structure is important for successful sequencing, but it does create some built-in insulation, which leads to longer thaw times. And the last point that I want to make on this is that it is important to follow the thawing guidelines on the iSeq 100 system guide to allow for adequate thawing, as it's crucial for a successful run. We want to make sure that those reagents are fully thawed before we start that run. Last, there's not really a way to make it go quicker than this. Our internal labs, have tried thawing these at warmer than room temperature water baths, but it can cause quality declines and cause clustering failures. And so do recommend, thawing at a room temperature water bath, no warmer than that, or it can cause some errors there. All right, and then the next thing after we're done thawing our reagent cartridge is diluting our libraries. We'll dilute that library and load it onto the cartridge itself in that nice orange placeholder for it. Successful sequencing does depend on that library being fully constructed and at a proper concentration. In this step here, we have adding PHYX. It is optional but recommended, as it is our standard positive control library. In general, for a whole genome library, we recommend 1% PHYX spike-in, but you may have to go up to as high as 20% spike-in, just depending on that library's base diversity. If you have any questions about that, feel free to reach out to us in technical support. We're happy to kind of go over your library composition and recommend, certain PHYX concentrations that might, or PHYX spike-ins that might help get us to a successful run. And as we talked about earlier, the system guide does provide some guidance on the dilution and final loading concentration. For a few of those library types, the system guide gives a recommended loading concentration. If your library type is not in that recommended loading concentration, we recommend starting at 50 picomolar and then optimizing from there. The loading volume for all library types is always going to be 20 microliters. And again, we're going to be loading that double-stranded DNA, and then the instrument will do that denaturation for us. All right, and then the next thing that I'm going to show you is a video resource that kind of goes over sequencing and how to start up a run on the iSeq 100 instrument. This is a user starting a run from a local run manager run. And so we can go ahead and go over and take a look at this video resource here. So first you start up at that sequence screen, the user presses sequence, and immediately it starts prompting you to load the cartridge. It recommends that you invert that thawed cartridge 5 times. It's really important to make sure that we're fully mixing the reagents in that well, and then subsequently tapping the reagent cartridge down on the lab bench to make sure that all of the reagent goes to the bottom of those wells. We're next going to go ahead and load our library onto the cartridge. We're then going to insert the flow cell into that cartridge, and then we are going to turn it around and insert it flow cell first. Unfortunately, it's not like a VHS, so you can't place it in there and push it in. Once you place it on there, you go ahead and click close door, and then the instrument will load the cartridge for you. Here we can see our user is looking for their run that they set up in Local Run Manager. We can see that they have 3 runs available to them. This person's going to go ahead and start demo run 1. They can double check that the reads and cycles look good, and the output folder looks good, and then it's just start run. Immediately from there, we're going to go into our pre-run checks. Our pre-run checks are set up into 2 different sets here. We have the instrument check, which is kind of all of the dry checks on the instrument. And then we go into the flow check. Once we're in the flow check, the instrument does actually pierce the reagent cartridge and start to flow liquid over that flow cell in order to test its usability. If the system checks do fail during the flow check step, unfortunately, we will have to get a new reagent cartridge to start the run up again. But if everything goes according to plan, we'll go into sequencing. And here you'll get an estimation for when that run is going to complete, as well as the projected total yield and the Q30 metrics for the run here. This is just a demo set here, and so the Q30 for read 1 and read 2 are pretty immediately available. In reality, again, about 5 hours after that run starts, you'll start to see the Q30 metrics for read 1, as well as some projected total yield. Once this run goes to completion, you'll get a screen that lets you know that that run is completed. It'll give you the high-level metrics. It'll tell you when the run completed, and then it will give you the option to eject your cartridge. And that's really all that needs to be done. Once you eject that cartridge, you can leave the instrument as is and get ready to sequence again. And then you'll go back to that home screen there. So moving on to what happens after a run completes. So again, those basic run metrics are displayed on the screen, showing if that run meets specifications. That slide that I provided earlier can be a good rule of thumb. Was this a good run? You can use that resource there. And then, if everything looks good, you can, continue on with data analysis for that run. You can, after you eject that cartridge at your convenience, you'll just need to discard that kit. For reagent disposal, we do have our safety data sheets on our website, linked here at that support.illumina.com site. And again, just an important note that the flow cell is considered electronic waste because of that CMOS chip. And so it will need to be disposed of accordingly. And then that information in the SDS sheet will help determine the best way to dispose of this reagent cartridge according to your institutions and local regulations. All right, and then moving right along to run monitoring and evaluation, there are three main ways to evaluate a run. The first one is, again, on that instrument screen that was just shown. You can also take a high-level look in Local Run Manager to see how that run looks. And then we also have Base-Based Sequence Hub or Sequencing Analysis Viewer on instrument. The on-instrument and Local Run Manager is a great high-level overview of, is this run looking good? While Base-Based Sequence Hub and Sequencing Analysis Viewer will provide some greater insight, such as the % occupied metrics, how much library actually bound to the flow cell, as well as things, like how much Bi-X is aligning on the flow cell and things like that. And so if you want to do a deeper dive into the run metrics themselves, highly recommend using one of those resources. And then as far as instrument maintenance, the iSeq 100 instrument really does require minimal maintenance. Again, we're not washing this instrument necessarily. Occasionally, runs will need to be deleted from the instrument. We need at least 2 gigabytes of free drive space in order to start a run. In order to delete runs on the instrument, we recommend using Process Management, which is a tool in the settings of the iSeq 100. As we talked about, like with the V2 control software, if your instrument's connected to the internet, the instrument will let you know when a software is available. And so you can go ahead and if that software update's available, you can click a button on the iSeq control software that will start that update for you automatically. Or you can manually download that control software and set it up on the instrument and run it manually. And the last thing that you'll need to do is replace the air filter on the iSeq 100. We do recommend replacing it every 6 months or more often if you're operating in a particularly dusty environment. If there's any construction in the lab or in the lab next door, it might be helpful to replace it earlier. It does take about 10 seconds and does not require any tools. We have some guidance on replacing that air filter in the site prep, or not the site prep guide, the system guide as well, if you have any questions about that. All right, and so that's kind of the workflow of using the instrument. And now I kind of want to go over a couple of tools that we have for any unexpected situations that might come up. The iSeq 100 has a really great resource guide within the actual system guide itself in Appendix A, Troubleshooting. It has a lot of the errors that we might encounter on a run, as well as some ways to resolve them. I love this instrument troubleshooting guide here. It really goes through kind of the first check that we would do in tech support. I do want to note, this is some guidance on how to do some troubleshooting, but as always, feel free to call Illumina Technical Support. We're here to be able to try to help you make sure that you're getting the most out of your instrument. And so, as always, feel free to give us a call so we can try to help troubleshoot some of these things. But if we're not available for whatever reason or it's not convenient, we do have some guidance on how to resolve those errors. So, the basic workflow is you get an error on the control software. The first thing we recommend doing is power cycling the instrument. Power cycling is a little bit different from just shutting down the instrument or restarting it. The next slide goes into detail about what that power cycle actually is. You'll hear a lot of us in technical support use the term power cycle, and it generally means a pretty specific way to shut down the instrument. Once you turn the instrument back on, if that system initializes, we recommend performing a system check using that test cartridge and test flow cell. If the system check passes, we would recommend continuing to use that instrument because we then know that the instrument itself is running well. If it fails to initialize or the system checks don't pass, please do give us a call. We can take a look at those logs on the iSeq 100 and take a look at what might be going on that's getting us to continually run into these errors here. And then that's the basics of the troubleshooting. So let's dive a little bit deeper into what that actual power cycle is. This is tech support's most common and generally most effective tool that we can use while doing some remote troubleshooting. And essentially, it is shutting down the instrument, switching the power off with the rear toggle switch. This part is important because it allows the instrument to completely turn off allowing all of the subcomponents to lose power and go to a home position. It's also important to wait at least 30 seconds. It's nice to have like maybe a lab timer there, or if you have more time, maybe grabbing a coffee and then coming back before turning it on. Once that timer has expired, you can go ahead and switch the rear toggle switch on, and then you should see that power button start to glow. You can press that power button, and then it'll turn on the actual computer, and then that iSeq control software will initialize after you log in. And so whenever you hear a technical support rep mention power cycling, that's generally the workflow that they're talking about, is this shutdown, physically turning the instrument off, waiting, and then bringing it back up. The next tool that we have, if you suspect there are any hardware issues with the iSeq 100, is a self-diagnostic test called the system check. This can be run, again, with that same test kit that's used during the first time setup, and it is a comprehensive check of every subsystem on the instrument. If a system check passes, we know that the instrument is working properly. If you ever get a system check that doesn't pass, again, give us a call or email tech support, and we'll be able to review the logs, determine the next best steps. That reusable test kit can be used up to 130 times, and the software will let you know how many tests are left when it's used. The total time for a system check is about 45 minutes from start to finish. And we have some guidance on how to do that system check in the user guide there on page 46. All right, and then last but not least, the last feature we have on the iSeq 100 is a factory restore. It's one of the best features, I think, on the iSeq 100 because, if there are any potential configuration issues with the Windows computer, we can go ahead and bring it back to a factory restore and see if we can get a resolution that way. As a note, only the C drive is wiped, so all of the information on the D drive, any of the data that the lab produces, is left untouched. After a restore, because that C drive is wiped, however, the control software, the local run manager, and any associated modules like Generate FastQ or any Amplicon analysis modules will need to be reinstalled. It only takes a couple of minutes to do that installation, but I did just want to note that you will have to do manually reinstall all of those softwares. Every time the iSeq 100 is booted up, this screen is going to show. You all have probably seen this screen if you have an iSeq 100 in your lab. It shows for about 5 seconds, allowing you to choose to restore if you'd like to. It is normal, and you can either tap iSeq 100 or ignore it if you don't want to restore, and it will automatically boot into the iSeq 100. However, if you or someone in the lab accidentally hits that restore to factory settings, it's not a big deal. I have had a couple of calls where that has happened. Restore to factory settings does take about 30 minutes. During that time, the instrument will start and restart a number of times as it goes through that factory default. And then again, the only thing that you need to do once it comes back up is reinstall the control software from the website. And so that is kind of like the last tool that's really helpful for troubleshooting I wanted to highlight here on the iSeq 100. And next, we can talk about all of the great resources on the iSeq 100 instrument. So the first thing I want to highlight is just our general support page. There are a lot of different resources here on the iSeq 100 support page. And so if you're just kind of interested in, what is available, this might be a great resource just to see the plethora of different resources that we've got on this support site. For some more targeted, search approaches, I've highlighted a couple of the ones that I find helpful and that you might as well. The first one, again, is that iSeq 100 sequencing system guide. It contains a lot of great information, including, how to do the dilution, how to do the loading. It includes guidance on the maintenance and the troubleshooting steps that we just talked about in that last section. All of that is in this sequencing system guide. I think this iSeq 100 system sequencing guide is one of the most comprehensive guides we have on an instrument, and so there's a lot of great information in here. I personally like to bookmark it as a web page rather than downloading and printing out the PDF, just because this does get revisioned every once in a while, especially when a new control software version comes out. And I have a link to that system guide down here. Next up, just wanted to highlight some of our web-based trainings on the iSeq 100. I think that particularly I like the Does My Run Look Good? We'll talk about Illumina Proactive at the end of this one, but discovering the benefits of Illumina Proactive is a really helpful one as well if you have any questions about how Illumina Proactive could be used in your lab. So there are some great web-based trainings here, and I also have the full list there on the bottom of this screen. Each of these is like 15 to 20 minutes long, and then they really, watching all of these can help you become an expert in the lab on that iSeq 100. Next is the Sequencing Systems Site Prep Guide, and I really wanted to highlight this one as a great resource, one, just for figuring out a great place to set the iSeq 100, but two, it's a really great place to work with your IT team on how to network this iSeq 100 Windows 10 computer. I highly recommend reaching out to IT, and if you are going to send this on a network or put this on the internet, working with them to make sure that it is managed appropriately. Again, that iSeq 100 does run on a Windows 10 computer, but we don't want to treat it like other Windows PCs that might be actively managed on a network. And the Site Prep Guide discusses, how we recommend the iSeq 100 to be managed. It also has some guidance on setting up that network. If you or the IT team has any questions, feel free to send that along to techsupport@illumina.com or give us a call. Happy to kind of go over your particular situation on the networking side of things. Next, again, I've already linked it 3 times already, but I wanted to just highlight again this CMOS chip and one-channel SPS chemistry. I think one of the really cool parts about this is that it does compare some run metrics from a mini-seq and a MiSeq, just to kind of show how that runs on the iSeq 100 instrument. We also have a lot of great product literature about different applications being used on the iSeq 100. I particularly like the sequencing library QC with the iSeq 100. It kind of goes over how to use this instrument as a QC machine before it goes on to a NovaSeq instrument. I also think the 16S metagenomic sequencing on the iSeq TechNote is very, very helpful to kind of go over how that application might be utilized on the iSeq 100. And if you're interested in how a run looks on the iSeq 100, check out the public data sets here. We have some public data sets on basebase.illumina.com, and from there you can download and review some run results or some run metrics, as well as some actual analysis results as well. So a great resource there. And last but not least, the last resource I wanted to share with you is Illumina Proactive. It's a great tool if you choose to connect to that Illumina Proactive portal. Instrument metrics and performance metrics are sent to Illumina, and it can alert us of certain issues on the iSeq 100. Here we have some links to the Proactive support page, some overview, some tech notes, as well as some FAQs that can provide you with some more information about what is actually shared with Illumina. At a high level, we're not getting BCLs, we're not getting sample names, and we're not getting FASTQ files. It really is just, Illumina iSeq 100 hardware metrics and the run metrics themselves. We also have a plugin with MyIllumina, if you have a MyIllumina account, that allows you to review the historical trends of that iSeq 100, so you can kind of see how the instrument has been running and been used in the past, which is really helpful as well. And it really just helps efficiently perform some troubleshooting steps instead of, myself or one of my other colleagues asking you to collect run metrics and send them over to us. We can just review them on the spot. And we can also, use some of that instrument hardware metrics to try to catch issues before they actually arrive. All right, and so thank you all so much for listening to all these different resources. I really appreciate your time. Before we